Alex, you. Yes, he is. Okay, hello everyone. Welcome to this month's Heart Muscle Talks on behalf of the International Cardiomyopathy Network. And today we're very pleased to welcome our colleagues from the AMC in Amsterdam, Connie Betsina and Sean Jurgens. Connie, welcome. Sean, welcome. Connie, perhaps you'd like to introduce Sean and maybe the work that we're going to be hearing about today.
Sure. So it's a pleasure to introduce Sean Jurgens. Sean is a talented PhD student who's in the final stages of defending his PhD. So he'll be defending his PhD in October. And he's conducted his PhD across our group in Amsterdam and the group of Patrick Eleanor at the Broad Institute in Harvard. And Sean will be presenting our most recent work, which is a large meta-analysis of dilated cardiomyopathy and a whole host of downstream analysis on the data. And I hope you find it interesting. Sean.
Fantastic. I look forward to it. And I'll be co-chairing today with Luis Lopez, and we'll be taking questions after the presentation. So over to you, Sean.
Cool. So can everyone see my slides?
Absolutely.
Perfect. Well, thanks for this nice introduction. And indeed, I will be talking to you about our work on a large genome-wide association study for dilated cardiomyopathy. So probably this background is quite basic for many of the people watching, but I'll give a short background in dilated cardiomyopathy, or DCM. It's characterized by increased ventricular size and a decrease in contractile function that is not sufficiently explained by coronary artery disease or abnormal loading conditions. Prevalence ranges are, well, they range a bit, but maybe 200 to 400 people can be expected to develop the more idiopathic form of the disease in their lifetime. And it's an important cause of heart failure, arrhythmia, and sudden death, and is one of the leading indications actually for heart transplant. So it's a disease that's important to be understood, to understand the molecular mechanisms. And one of the approaches that has been taken to understand the disease is by genetic analysis. So we actually know from past few decades that rare genetic variants are an important cause of the disease. Rare causative variants are found in maybe 25, maybe 30% of cases, but most probands are not found to have a monogenic variant, so rare causative variant. There's a lot of heterogeneity in the severity of the disease. So some people carrying a variant might never develop DCM, or some might develop a relatively mild form, and others might require heart transplant early in life. And actually, a lot remains unknown about actionable causal pathways that influence the disease but might be targetable. So other approaches to understand DCM and the molecular mechanisms through genetics have been by genome-wide association studies. I'll show you a little bit more of the background of those in a second. But what these previous studies have shown, a few have been published, is that there's a substantial common variant heritability. Common variants contribute to the disease. But given the large sample sizes required for GWAS, only a handful of loci have been identified as of yet. So what do we do in the GWAS? This is just for the people who might not be as familiar. Essentially, in this case, we take a whole lot of patients and we compare them to a whole lot of controls. And we essentially take like the common variant fingerprints, a few 100,000 to a few million common genetic variations, and we compare them between patients and controls. And then you get this screen from the beginning to the end of the genome, where everything above the dotted line is statistically enriched, or either in the cases, the patients or the controls. And then another concept we'll introduce here is an MTAG, is where you essentially take this first GWAS and you combine it with another GWAS. So for instance, you could be looking at a GWAS of cardiac MRI traits, and these have been published. Think like ejection fraction or volumes. And by statistically incorporating these data, you can create another GWAS, which is hopefully better powered, and we call this an MTAG. So this is just an important another concept, is like a boosted GWAS, where you integrate the GWAS with other traits. So in our case, we aim to take DCM GWAS from various sources. So first we queried various biobanks, and we try to define this stringent phenotype of a non-ischemic DCM, which was a I42.0 billing code for dilated cardiomyopathy, but also excluding any antecedent acute coronary syndromes or revascularizations to try to remove coronary disease as a cause. And the three main biobanks we looked at were FinGen, UK Biobank, and Mass General Biobank, totaling over 5,000 cases. And we also had these clinical cohorts, case control cohorts from health systems, one published in Garnier et al., one published by Mader and colleagues, and a new data set from Amsterdam with 783 cases. And these all were clinical criteria cases with imaging compared to healthy controls. And those were approximately 4,300. By combining these data sets, we got a data set of 9,365 cases and almost 950,000 healthy controls. And as a spoiler, in this large meta-analysis, we attained 38 significant loci, which I will show in a second. But then we also have the MRI traits for the MTAG I just explained. So we took various approaches to find the cardiac endophenotypes that were most strongly associated genetically with DCM. And those ended up being, well, it's actually a typo here, but it was the global circumferential strain. It was an MRI parameter with one of the strongest correlations with DCM on a genetic level. And also the left ventricular end systolic volume index to body surface area. So we integrated those two GWAS for those two traits that have been published and integrated them with our GWAS to make this what we call the MTAG DCM, which uncovered substantial, additional loci. So when we look at this Manhattan plot, this is the screen from beginning to the end of the genome. And again, each of the dots above, or each of the towers that go over the dotted gray line are significant loci. And we've annotated them here with a likely causal gene that's nearby. And the most notice, so I'll go into some of the genes later. The bold genes here are all novel loci. And indeed, it turns out that we have 27 novel for DCM. Based on what is now published in peer-reviewed journals. Then we mirror the Manhattan plot to show the MTAG, and we actually go from 38 to 65 loci, of which 50 are novel. And taken all together, there were 70 distinct loci across GWAS and MTAG. This is our gene prioritization, because of course, what I just showed you is a huge screen, and we're just saying, well, this is a locus, but what is the gene? I won't go into detail what all the methods are, but we used here 5 different methods, or 6 actually, different approaches to assign points to genes near these peaks. And everything here in dark blue or light blue essentially means that the gene on the y-axis attained a point from the method, or on the x-axis attained a point from the method on the y-axis. And it's ordered from most prioritized on the left to least on the right, and most on the top, and then slightly less on the bottom part of the figure. So when we look at the left here, the most left gene is BAG3, and BAG3 was prioritized by almost all of our methods, and that is a known Mendelian cardiomyopathy gene. And if we then query which of these genes are known Mendelian cardiomyopathy genes, actually there's a striking enrichment in our highly prioritized genes for Mendelian cardiomyopathy genes. So we have BAG3, FOD3, FBXO32, Titin, Actinin2, CSRP3, ALPK3, phospholamban, filament C, and more. Just showing that our approach is nominating a lot of known genes for DCM and other cardiomyopathies, but also a lot of other stuff, of course. That's the important thing. We also want to find new biology, and there's loads of targets being prioritized here. So if we think about some of the interesting novel targets, you can look at SVIL, which is an actin-binding protein implicated recently in hypertrophic cardiomyopathy. We also have NED4L, which is a ubiquitin ligase implicated in model organisms in sodium channel regulation, but also heart failure. We have MAP3K7, a mitogen-activated protein kinase implicated in cardiospondylofacial syndrome. TMM1A2, a regulator of myoblast differentiation. And strikingly, multiple genes involved in cellular stress responses, notably the heat shock stress response pathway, which is very striking. And this also includes HSPB7, which is being validated by various groups around the world now as having an important role. in cardiomyocyte homeostasis. So the next thing we wanted to do is take these GWAS data and these prioritized genes and query single cell data. So luckily, there's a lot of data sets published over the past few years. And we use these three. So one from Chafin et al. In Nature, one from Koenig et al. In Nature CBR, and a paper by Reichhardt and colleagues published in Science. And we harmonized these data sets, which were all single cell or single nucleus RNA-seq. And this gave us 142 left ventricles, or data from 142 left ventricles, of which 61 non-failing and 81 end-stage DCM hearts. So the first thing we did is we wanted to query cell type enrichment. So we wanted to say, can we say from our GWAS data that heritability is enriched in certain cell types, in genes expressed in certain cell types? And here we see results from the Chafin dataset, where we see different cell types on the x-axis and the significance on the y-axis. And most notable is that heritability for cell type enriched genes was very strong in cardiomyocytes, strongly enriched, but not really in any of the other cell types, notably. When we then looked at the second data set, Reichhardt et al., this was even more overwhelming. So we actually saw only enrichment for heritability in the cardiomyocytes and not in the various other cell types in the single nucleus data and single cell data. This is striking because when you look at the observational data, so when you look at the cell type changes that happen between DCM and or between non-failing and DCM hearts, all the cell types show something differential. So all the cell types are different between the non-failing and the DCM. Yet based on our GWAS data and based on our heritable component, we cannot recapitulate that, showing at least indicating that cardiomyocyte dysfunction or the intrinsic function of the cardiomyocyte is a very important initiator of the disease. Next, we wanted to look at specific genes in specific cell types. So here we just focus on some of the most highly prioritized genes. And we plot these genes here in a heat map on the x-axis and different cell types from our single cell data on the y-axis. And here, essentially, the color of a dot indicates the relative expression of that gene in the cell type, and the size of the dot indicates the magnitude of expression. So if we look in the bottom right here, we're looking at cardiomyocyte and titin. Well, titin is super red, so it's very strongly expressed in cardiomyocytes. It's got a large dot, which also indicates high expression. And this is consistent with what we know, that titin is highly expressed in cardiomyocytes. And one of the striking features we see is that many of the prioritized genes are preferentially and highly expressed in cardiomyocytes, although some genes are more widely expressed. When we then look at which genes change between DCM and non-failing hearts, those are the blocks, so the black borders. And we can actually already appreciate that some genes like CAMK2D, phospholambam, HSPB7, and more are significantly differentially expressed in our single cell data between DCM and non-failing hearts. So to kind of summarize, we intersected our GWAS with single cell data, and we first showed that the main heritability of DCM on a cell type level is enriched in the cardiomyocyte. And then when we look at what targets are preferentially expressed in the cardiomyocyte, some interesting targets emerge, like MLIP and PDLIM5, which are predicted to be involved in the contractile apparatus. With MITF, a transcription factor previously implicated in cardiac hypertrophy. We have ERBB4, the tyrosine protein kinase implicated in heart development, but also many others that have a less clear role in the heart, like TANK1 and CRIM1. And then we saw several genes that show a significant difference between the DCM expression and the non-failing expression. And those, again, recapitulate the heat shock response. These were all differentially expressed, but also MTSS1, which is in the shear stress response. So these are all interesting targets potentially for further validation. We have CAMK2D and PRKCA, which are involved in calcium handling, and also phospholambam. So now we've done the target validation, and we want to do other downstream work. So we want to say what other mechanisms and what other causal links between traits and DCM can we find? So to this end, we use Mendelian randomization, where you essentially want to say you have a GWAS for an exposure trait, you have a GWAS for an outcome trait, and using genetic variance as a instrumental variable, you want to say what is the causal link between the exposure and the outcome. This is Mendelian randomization helps us answer this, although it should be noted that it has a lot of limitations and a lot of assumptions. And I won't go into all of them at the moment, but some of the assumptions are essentially that the instrumental variables do not associate via confounding variables, and that the association is mediated entirely via the exposure. I won't go into all the other assumptions. The point is, this is the Mendelian randomization can help us get clues about causality, and I'll show you some results, but they always have to be taken in the context of other literature and other data. Anyway, these are the results from our MR screen, where we looked at 75 potential risk factors for DCM and their association with DCM in a Mendelian randomization framework. And everything here above the red line or below the blue line is significantly associated in our screen. And we find no traits associated with lower risk of DCM, but we find several potential causal risk factors for DCM where the risk factor increases risk of DCM. So we see that increased body weight, increased, atrial fibrillation, increased body mass index, increased height, and increased systolic blood pressure are all associated with increased risk of DCM in our Mendelian randomization screen, and therefore potentially suggests a causal role for these traits. Conversely, we turned it around. It's a bidirectional screen, and satisfyingly, we found that DCM was causally associated with increased risk of heart failure. Well, we know this. That's great to see. And we saw a signal for lower platelet volume. I should note that most of these signals survived all our stringent sensitivity analyses, except the platelet volume outcome, which is nice because we couldn't quite explain it. And that one was lost in our sensitivity analyses. So when we focus back on specifically weight and systolic blood pressure, which are obviously modifiable, we could see that across different methods for Mendelian randomization to try to account for the different assumptions, we see a consistent signal in this forest plot where genetically predicted weight and genetically predicted blood pressure remain significantly and robustly associated with the risk of DCM. So now we move to a polygenic risk score. This is where we integrate all the genetic data, all the risk loci we found into a single continuous score, which we try to use to predict DCM and other traits. So we made this risk score, and this is what a distribution can look like in an independent set of patients from the AMC. And we see here in red the distribution of the polygenic risk score, the PRS, in DCM patients versus general population in gray. So you definitely see some separation, obviously not perfect, we don't expect that, but we see some separation in the distribution between DCM patients and controls. And when you look at this forest plot, which shows the effect size for DCM per standard deviation of the score, we see there's actually quite a strong score. So per standard deviation, there's about an additional 90 or 0.9 or almost twofold odds per standard deviation per standard deviation of polygenic score. And this was consistent in people of female and male assigned sex at birth. Then we questioned whether the PRS might have a different effect depending on whether the probands carried rare genetic variants known to cause DCM. And we did see a differential effect. So here we see the forest plot again, and red are individuals carrying class 4 or class 5 variants for DCM, and blue is comparing genotype negative, so patients who have no known or potential causative gene variant for their DCM. And indeed, we see that the genotype positives show a smaller effect size, odds ratio around 1.5 versus 2+. But still, the PRS is significantly enriched in these DCM cases. So this tells us that the PRS contributes to the DCM liability in both genotype positive and negative cases, but that the PRS, well, that cases need more PRS, more polygenic burden to reach the DCM state if they don't have a rare variant, which makes sense. And here we also show the distribution, where indeed you see that the genotype negative cases have a distribution of the mean moved the most to the right. So they have the highest polygenic score. And the genotype positive have a mean PRS that's kind of between the controls and the genotype negative. Lastly, for the PRS and DCM, we looked at the association in different ancestry groups. And to this end, we used the All of Us data set, which is this very diverse data set from the States. And essentially, we would show that the PRS was associated with DCM in African individuals, in European individuals, and in Hispanic and mixed American individuals. Although we could identify the strongest prediction in terms of odds ratio, but also other parameters in the Europeans. And this is what we would expect because our GWAS was mostly European. So what we can tell is the PRS does work to an extent in all ancestries, but it still works best in Europeans as expected. Finally, we looked at the DCM PRS as a predictor of more broad systolic heart failure in all of us, which obviously is a much more common disease and might be diagnosed in the context of a lot of different primary diseases and risk factors. And here, essentially, we looked at whether the DCM PRS can predict systolic heart failure in different settings. So the first is in patients who have a diagnosis of prolonged hypertension. And indeed, stratifying by tertile, we could see that people with an increasing DCM PRS had increasing risk of systolic heart failure after their hypertension diagnosis. The same in patients diagnosed with AFib. There's a significant increase in systolic heart failure risk after their AFib diagnosis based on DCM PRS. But even after myocardial infarction, even though the effect sizes are lower, we could see that individuals with a higher DCM PRS had more systolic heart failure after their myocardial infarction. So I presented a lot of data. I'll give a small summary. In this study, we assembled a large-scale DCM GWAS and MTAG for gene discovery, and we found 70 susceptibility loci, 63 prioritized genes, and then when we intersected these GWAS data with single-cell data, we identified the primary initial role of cardiomyocytes in the disease, but also highlighted cell type-specific pathways and gene prioritization. We constructed a PRS for DCM, which associated with DCM across ancestries in genotype positive and negative patients, and also predicted systolic heart failure across various settings. Finally, in a Mendelian randomization framework, we investigate potential causal links between DCM and other traits, and found that there's a potential actionable component in higher body weight and higher blood pressure. So these results may inform further studies into mechanisms underlying DCM and myocardial function. Because these GWAS hone in quite strongly on this cardiomyocyte, on the contractile function, it might be a very good target to find mechanisms that underlie specifically the myocardial function, but also, of course, a therapeutics discovery associated with that. So finding therapeutics that might affect contractile function. And finally, the PRS and the Mendelian randomization results might inform clinical management of DCM patients or at-risk family members or potentially even individuals who were found to be at risk through future screening efforts. So I will conclude with this being a team effort. I am presenting these data as part of huge collaborative efforts. And that's why I always wanted to show this author list, because lots of people contributed and would not have been possible without a lot of people contributing data and contributing ideas. And I want to thank all the co-authors, and especially, of course, Connie Bedzina, the PI of this effort. And I will have to also thank the Amsterdam UMC and the Hartstichting for providing grant support and personal support for my work, including this manuscript. Thank you.
That's great. Thanks, Sean. A really clear and interesting presentation. So perhaps, Louis, do you want to lead on the questions? And I'd encourage the audience to either post their questions in the chat, or as Alex says, please raise your hand if you want to join the discussion. So over to you, Louis.
Thank you. Thanks, Sean. Really, really interesting data. that builds on previous work. So it's very, very nice to see this building up to this stage. I don't see still any questions. Maybe just to go for a start on the discussion. I have a couple of questions. So one would be if you did also, so the same sort of unfolding between genotype positives and negatives that you did for the PRS, if you also did that with the Mendelian randomization for the weight, blood pressure, and so on. And the second one would be if you actually tried to go even further in the unfolding and tested for specific genes for the effect of the PRS. So I'm thinking basically of titin versus others because you know of the possible higher effect of acquired risk factors for titin, for example.
Yeah. So for the great questions, and for the first was whether we could look at genotype positive versus negative, for instance, in the Mendelian randomization. I think it is a very good suggestion, or I would be very interested in those results, although it is quite difficult at this stage to do that, because what it requires, the Mendelian randomization, as you know, is it requires that we have the GWAS for an entire GWAS for genotype positive and negative patients, right? And the issue is that for a lot of the data sets that we are using, we do not have that detailed information. So let's take the biobanks, for instance. For FinGen, we just don't know who will be genotype positive and negative. For UK Biobank and Massachusetts Biobank, we would have to go into the sequencing data and manually curate each and every variant in each and every case to decide who is positive and negative, and that is quite an extensive task. And then for several of the clinical cohorts, we just don't have those data either. So the only clinical cohort where we have those now robustly curated is Amsterdam UMC. So it will be a lot of extra work, but I think it's definitely something we could look into. It's just the thing that for an MR, you need a well-powered GWAS. So we need really well curated numbers for these genotype positive and negative, and we just don't have them.
Sure. And about the individual genes?
Yep. So we are looking into that. We didn't put it into this paper, but I can give the sneak preview that the pattern follows as you might expect. So we stratified now in some initial work the genotype positive into titin versus, well, malignant genes, which is like lamin A, BAG3, RBM20, and phospholambam, I think essentially those. And we found that carriers in those genes had PRSs that were very close to the general population. And the titin were closer to the genotype negative. And that was nominally significant, that difference. So you kind of see this gradient where you have the genotype negative of the highest PRS, then you have the titin positive, then you have the malignant gene positive, and then you have the general population. We even have some data from family members where we see that family members, for instance, who carry a lamin A variant but are not yet phenotype positive, have a strongly negative PRS compared to the general population. So they are somewhat protected, which is fascinating, of course. But these are all very preliminary data, and we still have to like validate it and make it more robust and increase the numbers.
Sure. No, that's very exciting to hear. It's good when things match our sort of concepts, right?
Yep.
Yeah, very exciting. Okay, I think we have one question from the audience. Are you able to read it, or do you want me to?
Oh, I can have a look. Sure.
Thanks.
As we are developing therapeutics for rare genetic cardiomyopathies, do you think that the PRS score can be used to identify individuals at higher risk for developing the disease process and thus being able to dose non-symptomatic patients earlier? Yeah, I think so. It's going to be, I think it will definitely depend on the gene, and we have to like validate the correct contexts. But I think definitely PRS could have a role in identifying the people who might be like eligible earlier for these kind of new therapies that are going to be coming soon. Yeah.
Well, can I just pick you up on that one? So mechanism. So it's always very interesting, isn't it, with this exercise? You've got lots of new hits, but the same old hits come up. Yeah, BAG3, titin, good old ALPK3, phospholamban. And in one of your slides, you very nicely sort of summarized the different, if you like, pathways that might be affected here. Because the heart, this is either hopelessly complex, or maybe it's not as complex as we think it is, because the cardiomyocytes have a series of core functions which relate to calcium handling, which relate to mitochondrial function, which relate to contractile function. So yeah, when we talk about targeting pathophysiology, are we talking about 5 or 6 of those final common pathways to disease, either alone or in combination? What do you think?
I think that is quite difficult to say based on only our data. So one thing I think is striking is that if I go back to the cell types, that there's a lot of hypothesis that all sorts of extra cardiomyocyte functions might be important in a disease like this. And I think they, like, for instance, fibroblasts or parasites might have some role later in the disease, but at least our data are not showing evidence, should say it correctly, but are not showing evidence that they have a very strong early primary role. And then when we look in this cardiomyocyte as the primary initiator, then we could maybe try to tease out a few of, like you say, I guess, these common final pathways that might be important. And we can identify a few based on our data. Also some others, like certain intracellular signaling pathways that might have been less obvious before. So I think there's definitely some new stuff there. Although I wouldn't want to go as far as to say, oh, our GWAS don't show X, Y, Z, so it's not important, right? I think that you can't go that direction. But definitely, I agree that our data do show that some known pathways are just really important, and some known mechanisms are just really at the core of the disease. And that's also good to be aware of when we think of where do we spend our money for targeting, right?
Where would you spend your money?
Yeah. Well, based on this, I definitely would be less inclined to look outside of the cardiomyocytes especially for early targeting. And I would maybe go for some of the intracellular signaling or the tyrosine kinases that we find here that were not as robustly associated before. So those might be interesting. So we've now established they're highly expressed in the heart. They have some experimental evidence that they are involved in ion channel function and contraction in some model organisms like NED4L or ERBB4. I think those are really interesting ones we could look at. And I do believe that in the end, a lot of them will function via either intracellular stress mechanisms or just contractility. Yeah.
What about BAG3? Because your signal for BAG3 is pretty. Was it the most significantly associated? I think it was.
Yeah, it's one of the strongest.
So what are your thoughts about BAG3 in that context? Is BAG3 the final? Is it the orchestrator of cardiac function? Why does it keep coming out consistently across studies, and not just in hypertrophic phenotypes? It comes out in other phenotypes too.
Yep. No, I absolutely agree. So, yeah. So it is the strongest peak. Yeah. So we have it here. Yeah. I am not a functional biologist in that sense. So I might not be the best to ask this. I'm more of a statistical geneticist and biologist, but I would probably not argue that it might be like one of the very important regulators of one of these few very important pathways. And in this case, I believe BAG3 is most important in the stress response and the clearance of all sorts of. Oh, Connie, what do you want to add something?
Yeah, I can add a little bit on that. Sean, if I'm not mistaken, the top signal or in a very high linkages equilibrium with it is a coding or 1 or 2 coding variants, right?
Yep.
And actually, those variants, at least one of them has been targeted and studied, if I'm not mistaken, in iPS-derived cardiomyocytes.
Yep.
Yeah, it is very interesting that might go through coding variation. And the same signal also comes up in HCM with an opposite direction. So for sure, very interesting to really see what's happening there, because with such a strong association being involved into cardiomyopathies, yeah, we really need to dig more a bit in there, I think.
Yep.
Yeah. I'll bring Alex in. As he's had his hand up patiently. Yeah. I mean, my understanding. I mean, I'm even further away from the biology than you are, Sean. But it's BAG3 does a lot of things. Or it's involved in a lot of processes in contractile function, in calcium handling. That's why I just wonder why it comes out all the time, because it has so many functions that it's sort of. I mean, you used one of my favorite words, resilience, in your title of your talk. It sets the vulnerability of the cell. BAG3. But it is very consistent.
Yep, it is very consistent. Yeah.
Yeah, and we have many heat shock proteins also. Maybe that's
exactly.
part of, this homeostatic function.
Yeah, exactly.
I mean, that is honestly one of the most striking just convergence, or how do you say, like enrichments in our data, is just the heat shock and the related cellular stress responses. It's really striking in the GWAS. And the only Mendelian one of those was BAG3. We have now FBXO32 as well as a potential Mendelian gene for DCM, but that was only recently discovered. So yeah, the evidence for this is an important pathway is quite new, but very, very strong. Yeah.
Alex?
I'm always a bit confused when I read GWAS studies, because a lot of the things that come up on a GWAS, they don't necessarily always match, let's say, the genes we encounter, in our patient cohorts or in our clinical practice. And taking BAG3 here, for example, that does show to have one of the strongest signals, at least on the GWAS on the top. But it's by no means like the commonest kind of gene that we see in clinical practice. And for example, one of our bigger subcohorts in DCM is desmoplakin, but that rarely comes up in any DCM GWASs. And of course, I guess that means is that the ratio of the variation in the cohorts you study versus the healthy population is not as significant as these other genes. But what does that mean? And because in my mind that means that there is we are filtering out a lot of important genes just because they do exist in the healthy population, or what we consider a control population. And how can we optimize our methods to try and include more of those?
I think I understand your question, although I won't entirely agree. So the thing is that we're looking here at a common variant architecture, and the common variant architecture might differ quite a bit in terms of the genes and the level of significance compared to the rare variants, right? So no BAG3, Titan's a bit different, but no BAG3 pathogenic variant is going to have a population frequency of more than 0.1 or even 0.01%, right? That won't exist. Otherwise, that might be very rare founder somewhere, but generally those won't exist. So what we're looking at here is a BAG3 variant that is a risk factor, right? This is not a pathogenic BAG3 variant that is giving you a 100-fold odds of a disease, right? This is a BAG3 variant that is giving you a 50% increase risk of disease. And it's common. It's like a few percent or common population. So each day you take a stroll and see lots of people, every so many will have this risk allele, right? It's just a risk allele. And the same goes for the Titan, right? We have a Titan locus here, but these variants are totally different for the most part than the truncating Titan variants that will cause DCM from a rare variant perspective. And the interesting thing is that what you might encounter from common variant architecture needn't mirror in terms of significance the rare variants. And the reason for that is that for rare variants to be, as the way I see it, for rare variants to become like Mendelian, or gene to become like a Mendelian gene, the effect size of damaging variants needs to be quite large, right? And we know that, or it needs to be relatively frequent as a whole, like Titan gives you like maybe a 10 to 20-fold increased risk of DCM, which is much more than any common variant. When you look at the common variants, selection gives you something really strange, because the selection could make it that a gene that, for instance, let's take PPP1R3C. It's possible that this gene's function in DCM is rather like peripheral, and it's not a super essential gene in any respect. That means that really damaging variants that inactivate this gene, might reach a high population frequency. And then this peripheral effect that is not large from a rare variant perspective, but this peripheral effect might become significant in a GWAS, which on the other hand, when you look at BAG3, no missense variant that has a similar pathogenic effect could ever attain that population frequency of the PPP1R3C gene in this theoretical scenario. So what selection does in the GWAS is that it kind of balances things out in a way. So you can't really assign more biological intrinsic value to filament C as to PPP1R3C, even though we know, or the other way around, even though we probably know that it is the case based on rare variant data. But you can't say that for sure based on GWAS. All we know is that probably these loci are causal, and after a lot of searching, we might have found the gene that's causal, but we can't say much about the intrinsic biological magnitude of knocking the gene out. You can't make any predictions based on the GWAS. And this is where the rare variant association testing and clinical like rare variant studies are still really important to be harm, and we need to find ways to intersect the rare variant data with GWAS to find, for instance, new Mendelian genes.
We have
Okay, thank you.
more questions from the audience as well. Sean, if you
Oh yeah.
want to have a look from Eloisa.
There's a question, how about hypertension, confounding, question mark, to be excluded in future studies? Yeah, I think it's an interesting question. We did not explicitly remove hypertension, especially in the biobank studies. Although some of the clinical cohorts that we included in our GWAS were selected for not having hypertension, but it's a minority of our cases. It's always a bit difficult. We have a lot of discussions in the consortia, also, for instance, regarding myocarditis or well, you know, other risk factors. Also, for instance, in HCM, you know, there's a lot of discussion, should we remove people with hypertension from our cohorts, even though we know that it's very abnormal for a person, even with prolonged hypertension, to get like a wall thickness of over 16 or something, right? That's very rare. And probably in DCM, given the high prevalence of hypertension, I don't think it is reasonable to exclude it entirely, is my personal opinion. I think that DCM is probably a combination of various risk factors, including genetic and including things like hypertension. But obviously there's like, you cross the line, right? Somewhere there's the clear hypertensive cardiomyopathy that is phenotypically different to the typical DCM. It's just difficult, but I don't think that removing hypertension entirely is the right way to go, especially with the biobank data sets. It's just not doable. Does it make sense? Or do other people have other ideas?
Yeah, I think Eloisa wanted to join the conversation. Alex, is Eloisa live? Hey, ciao, Eloisa.
The question is, if you take data from polygenic risk score in hypertension populations, and we have data and studies, and then you compare the polygenic risk score in hypertensive patients irrespective of DCM with the data that you have obtained in DCM, could this approach, this comparative evaluation, help in eliminating or subtracting some of the candidate genes that you have shown in your studies? This is the first question. Second point is, if we study monogenic cardiomyopathies in the same cohort of cardiomyopathy patients that we want to test for GWAS or polygenic risk score, I think that they probably could confound the results. And I would probably, I'm not sure, but this is my idea, separate and distinguish cardiomyopathies without evidence of a Mendelian cause or gene or mutation, whatever, from those that appear either sporadic or without any evidence of a genetic background. This is just for methods. What do you think about hypertension? Is a big deal for us. We always ask ourselves in this patient, but does hypertension have a role? And if I look at the polygenic risk score for hypertension, can I eventually find the same indications, same genes, and same information, or hypertensive patients with cardiomyopathy show different values or data?
Right. So I think there's a few things you could dissect here. So the first is from a gene discovery perspective in GWAS. I can say there are methods we could use and to actually try to eliminate the effect of blood pressure or hypertension, for instance. So that's actually very interesting, and we could look into that. Those are very statistical approaches. And that is more for GWAS, right? To see, do we still get the same genes? My bias is that at the power we are at now, all these loci will stay the same. I don't think these will change, but it is possible that potentially some of these are affected by blood pressure mechanisms and they might be lost. I don't think so. But the other question is more from a clinical perspective, seeing a patient in front of you. I don't think a PRS polygenic score or a PRS for blood pressure or DCM is quite there yet on their own, not even together, to definitely distinguish like a hypertensive cause from a Mendelian cause. I don't think it's there. It should be noted that we even find rare Mendelian variants in patients where we thought they were hypertensive cardiomyopathies. It's less enriched, but even there we find them, right? So it's just not so clear cut in practice. And I don't see patients every day at the moment, but I don't think that the data is quite strong that there's any hypertensive cardiomyopathies where you can be absolutely sure that they don't have a Mendelian cause as well. But that's just my inclination.
But this is very important what you say, because, and I guess you're right in a certain sense, because it means that we are still not able to clinically distinguish a cardiomyopathy with an hypertensive background from cardiomyopathies without any hypertension.
Yep. Yeah, it is difficult. I mean, there are definitely differences you can see in terms of the structure of the heart, but those are just not 100% specific or sensitive as far as I know. But I'm not an expert on those imaging criteria and such. Yeah.
No. Barry, do you think that we are not able to distinguish these two conditions?
If I put my old-fashioned clinician hat on, I think intuitively, yes. I think we see things that don't make sense. So our diagnosis of these cardiomyopathies are that you have an abnormality which is not explained by the presence of another trait, such as abnormal loading conditions. But it doesn't mean that you can't have those two things in coincidence. Right. And hypertension is very common. It's 30, 40, 50% of our cohorts. And yet, and it's very common in the general population, but the overwhelming majority of those people don't develop dilated cardiomyopathy.
Yes.
So there has to be a genetic vulnerability in some people with hypertension, and that genetic vulnerability may be rare Mendelian, it may be polygenic, it may be a combination of the two. But I think if you've got long-standing, really poorly controlled severe hypertension, then maybe you get heart failure. But in the majority of cases, it must be about individual susceptibility.
Right. And just to add to that, maybe I wasn't clear, but I was trying to say very specifically, it doesn't preclude there being a Mendelian cause. I wasn't saying they have DCM, right? Because by diagnostic criteria, mostly they would not qualify if they've got prolonged extensive hypertension, right? Then they would not usually qualify for DCM, but that doesn't mean they don't have a Mendelian cause of their cardiomyopathy, even though it's not a strict like idiopathic DCM, right?
Yeah, I mean, Luis, do you want to draw parallels with hypertrophic cardiomyopathy? Because the same applies to HCM, right? And with hypertension, LVH.
Absolutely. I mean, it's everything that has been said in terms of the challenge, both clinically and genetically to distinguish applies. And I was actually thinking of also another very challenging scenario to us where some of these things might have a role, which is the athlete's heart, and particularly for DCM. So it would be very interesting one day to have some data on PRS on athletes, because again, like hypertensives, same people that are sort of submitted or practice exactly at the same level, one doesn't change the heart at all, and you have another individual where it's very dilated, even the strain is down and all of that. So it would be very interesting to have those kind of data as well.
There
But
was
definitely.
actually, oh, sorry, go ahead.
No, go.
I was just going to say there was actually a paper published in Circ, I think a few months back now, where they actually, it was very interesting. They looked in athletes and they were still young, most of them, I believe. And they actually looked at a rudimentary DCM PRS, so not ours, but a very basic rudimentary one. And they actually found that the endurance athletes with a higher DCM PRS had increased volumes, like significantly increased volumes, which is very interesting. Yeah.
It's the resilience
Right.
concept,
Yep.
isn't it? So it's whether it's hypertension or exercise or alcohol exposure or maybe pregnancy. It's how well can your myocardium resist that onslaught. Connie, you had your hand up there.
Yeah, it's not a black and white thing. So, you know, susceptibility, different factors contribute to susceptibility. And some may predominate in one group as opposed to others. But I think we shouldn't look at this as black and white. You know, because a person who has a high polygenic risk score for hypertension, but then has a low polygenic risk score for the cardiomyocyte part of the story might still be at the right balance. So I don't think it's black and white. It's within each individual, we have a mix of this. And I think the challenge is, you know, how will we be able in the future to determine the exact mix in each individual? And not only genetic, but also external stressors.
Lifestyle, etc. Yeah, I'm waiting for Alex to say artificial intelligence, but you know, so Alex.
Artificial intelligence. No, what I actually wanted to comment is I found very interesting so when you show the score and the control group and the DCM, and you can see there is a significant overlap. And what my question is essentially is that separation all that you know, genetics can tell us about it, and the rest is just environment? Or do you think, you know, by improving the methodology of developing a PRS, we can further separate the two curves?
Yeah, there's a lot to be won. That's a short answer. So
yeah.
Methodology will make a huge difference, and that is broadly threefold. Where I would say the first part of it is just better GWASs and larger GWASs. So we have here only 9.4K cases. I mean, it's still massive compared to, you know, like what small studies, usual like cohort studies will give you. But there is on the table potentially in future collaborations over 20K, maybe even 30K. Like when you combine all the data sets that are now being assembled and also parallel efforts to ours. And that's the first, right? Just more data just means better scores. The second is just the pure methods of the PRS. The method I used here is surprisingly good, even though it only uses like 15% of all the variants you see here, which is quite ridiculous if you think about it. It's only using 10% of the information here, theoretically. It's not really true, but kind of only 10% of the variance. And it's already giving this good a score. But we can also use methods that actually computationally can use the full 9 million variants that we have plotted here. And those will do better. There's ones that better take into account the functional annotations. And there's methods just published in Nature Genetics that I think we want to try out that improve it. And the third part is this very difficult aspect of trying to incorporate the common with the rare and the low frequency, right? Common GWAS approaches usually look at more than 1% frequency for variance, but there's this pretty unexplored area of between 0.1% and 1% that I call low frequency. And there might be some prediction gain from incorporating variance there. I'm not sure, it mightn't be much, but I think those are broadly the three directions that could definitely improve it a little bit. Yeah.
Sean, Connie, unfortunately, I have to go to another meeting now, so I'm going to hand over to Luis to sort of close the final 5 minutes. But thank you. This has been a really fascinating conversation. So thank you so much for joining us today. But Luis, you want to take us down to the close?
Thank you.
Thank you.
So I think we still have time for maybe the last question that I'm seeing on the Q&A chat.
Yeah. So there was, yeah.
Now, still
For
me.
BAG3.
Yeah.
Oh, sorry. Tell me.
Whole exome and whole genome sequencing are going now, the results that we are reading every day in new papers, they are going a different and the opposite direction. Very rare new disease genes and very rare new information that can potentially help us in increasing the number of genotype patients. So the question is how to integrate the emerging data on GWAS and polygenic score and whole exome and whole genome sequencing. Can we do something? Can we get some clinical advantage or benefit today, or should we wait for practical applications or suggestions or whatever?
Yeah, I think it's an excellent question, and I think there's very straightforward ways that these data can be complementary. So what I was explaining before was this is a large list of genes that likely have some involvement, but we're not sure how peripheral in terms of effect size or how central or how large biologically, you know, the magnitude is of each of these genes. But it's still a good list to start from. So one approach, and colleagues are doing this, is to say, assume that there is at least one additional true Mendelian gene among these genes, right? A new one. And then this is already a good short list. So you can say, let's say in our clinical exome and genome studies, can we find an enrichment of these rare variants when we look at just this candidate list, right? And this has gotten some interesting results. We got an email from collaborators from France who actually said, we have a family where we have a MAP3K7 variant that co-segregates with the phenotype in a multi-generational one. And they never understood if the gene could make sense. And now we find it in the GWAS, and they're suddenly like, okay, this could make sense. So I think there's definitely ways these can be complementary.
Okay, thank you.
Yeah.
Okay.
So there's, I think the Shihab, I think you Alex, turned on your mic, so.
Hello, everyone. Thank you so much for this rewarding webinar. My question is about how the overlap between the genotype and sometime in late stages of HCM, and so there may be an overlap of the phenotype, specifically by cardiac imaging, and not just the HCM, and sometimes with the ARVC, especially if it is biventricular. How would this genetic overlap would impact the management, especially the disease label or the exact diagnosis we are leading, and the management specifically in the rise of new therapeutic modalities and drugs for management of HCM, for example. So this would be my question. Thank you so much.
Yeah, I think a lot goes into that question that goes very far beyond this genetic study. But one thing I'll say is that I think, well, one bias I personally have is that I probably would say that ARVC and DCM are probably more similar on a genetic level, more similar than DCM and HCM. Although it is true that that late stage HCM can present as DCM, and I'd probably say that like if we develop very DCM specific therapies, they might have very adverse or strange effects in the burnt out HCM, but that's just an inclination, I'm not sure. But what we see from our genetics is that from a common genetic standpoint, and probably also from the rare variant standpoint, DCM and HCM are opposites in terms of the basic molecular, or the genetic and molecular mechanisms. So I could almost draw you the same Manhattan plot, say it's HCM, and then just say all the betas are flipped, essentially. So all the effect sizes are flipped. And that's not true for every locus. So it's a bit of a lie. But they are either associated with both diseases with opposite effects, or they are associated with only one of the two. That's essentially the main gist of this. And that is extremely interesting because of what you say, that this HCM can burn out and become a DCM-like phenotype. But I believe that is just essentially the heart being so far in its hypercontractile state, so far beyond its energetic like capacities, that it just starts to fail again. But I think from the original molecular mechanism is probably opposite from DCM, and I agree we should probably take into account the initial mechanism in therapy as well. But it's far away. It's far away. Does it make sense, or did I not answer the question?
No, it is very interesting. I mean, this sort of mirror effect that Connie also published in the 2021 Nature Genetics paper. It is again fascinating as well, and as you say, can have very practical implications in terms of therapies. So with this, we have to unfortunately finish. We are it's 5 now. Thank you very much, Connie and Sean, for your excellent contribution to this. And I suppose we will see each other again back in September for another heart muscle talk. Thank you very much, everyone.
Thanks. Take care. Bye-bye.